right? So in earlier class we had seen about the diseases of oil seeds, right? So this totally I will be dealing with the diseases of oil seeds, right? So I will be starting with the groundnut first. So in earlier class we have seen about the diseases of oil seeds with respect to pathogen, which are all not exactly minor importance, or with respect to our locations. Okay, our geographical location, it might be little minor, right? But with respect to major one, we can study the fungal pathogens, right? So just now in this I try to exclude the whatever the earlier we have studied, right? So okay, let's start with the DS of groundnut, right? So it will be having more of fungal. So here and there, the important one I have included. Okay. To check out the diseases of groundnut, right? So these are all the major diseases, right? From early and late leaf spot, then rust, then alternaria leaf spot, then stem rot or sclerotium wilt, then collar rot or seedling blight or crown rot, right? So these are all the synonyms. It is having collar rot, seedling blight or crown rot, right? Then dry root rot or dry wilt, right? Then peanut, stem and bud necrosis, or it is a viral disease, right? Then root knot nematode. So these are all the major diseases which we will come across the groundnut crop. So coming to early leaf spot, which is caused by Cercospora arachidicola, right? So how you are going to differentiate between early leaf spot and late leaf spot? So that is the major criteria you should follow whenever you are entering into the field, right? So to know, the name only indicates it is a very early in stage, right? So before 40 days or when you sow, so after the two leaf stage initiation, the plant start getting symptom. Okay, so in seedling stage only the infection will start with respect to early leaf spot, right? So how it will be looking like means usually the spots will be very large, right? And more prominently it will be seen on the upper surface, right? More prominently seen on the upper surface, and usually the size of the leaf spots are very large, right? Then it is more of uncircular, or you can call it as uneven, right? It won't be having high circular nature. Okay, so it will be little bit uneven in size. So these are all the, and moreover the infection or the devastation caused by the early leaf spot is more or less severe than late leaf spot. So these are all the criteria you should keep whenever you're trying to identify the early or late leaf spot, right? So see, the stage of infection is 1 month after sowing, right? So once the seedling starts, then you can see the infection of this particular pathogen, right? So of course, whatever the leaf foliage or whatever, so after defoliation, the kernel size will be reduced, right? So coming to late leaf spot, as I told there, it is caused by Cercosporidium personatum, okay, or Favoisorapsis personatum, okay. So in this, usually as the name indicates, it is infecting at later stages. See, after 55 to 60 days, okay, after sowing, usually it will start. Then what are the criteria to identify is, usually it is mainly confined to lower surface. There we have seen it is a more of upper surface. Here it is more of lower surface. Then it is more of black lesion, right? Actually, I forgot to tell you one more point. See here, usually it will having yellow halo. See, yellow region will be more prominent with respect to early leaf spot. But here it is more or less of, or you may not be able to find the yellow boundaries, right? So that is the differentiation character. No yellowing boundary, okay, and defoliation will be very high, right? Because it is coming at the later stages or the senescence of the plant, right? So during that time, the plant will have more defoliation than the early stage, right? And yeah, so usually you can see in the lower surface as the major criteria, and it will be more of circular in nature. and size will be little less when compared with the early leaf spot, right? So as the Sarcosporadium or Sarcospora, usually it is a conidial nature, right? So it will be like needle-like structure, and it will be of nearly 5 to 12 celled, right? So like that it will be there, and usually the conidiophore will be geniculate in nature. Geniculate is nothing but like this, okay? So like this it will be geniculate, angular nature. So on that it will be bearing. Okay, it is usually colorless, right? And sometimes the conidiophore will be colored. Okay, so these are all the characteristics of the late leaf spot, and you know that it is a perfect stage is nothing but mycospirilla, right? This is the perfect stage. So what are all the predisposing factor for this particular disease, right? So as you see, whatever the fungal pathogen, most of the fungal pathogen, okay, so it should not be generalized. So most of the fungal pathogen will prefer the high relative humidity. So you can see prolonged relative humidity for just for 3 days. It is more than enough, because you see that in Kharif, usually the relative humidity will be very high, right? So 3 days is very mere, so it can easily get. Then low temperature of 20 degrees Celsius, right? With an dew. So the dew timings also differ. Okay. Then leaf surface, heavy dose of nitrogen with respect to all diseases or the leaf diseases. Usually high nitrogen dose is there, means the predisposing of the pathogen will be high, right? So, and deficiency of magnesium. So all these will make an whatever tetrahedron you will call, and it will predispose the pathogen. Okay, so the infection will be very high. So if you put disease area under curve also, you will get high incidence, right? Okay, so okay, lastly I will deal with the management because so many slides are there, right? So coming to groundnut rust, so it is caused by Puccinia arachidae, right? So as the symptoms, what you can see means the pustules will be, it will be underside, right? So when you just turn down the leaf, you can see lot of brown colored raised powdery color mass. Okay, so if you just rub it, all it will be rust, just like a rust you can easily see on your fingertips, right? So like that it will be there, so you can easily identify. And because of this, there will be lot of defoliation at the end of season, right? So that is also another major problem which groundnut will be facing, right? So see, it can be easily identified based on the orange red color, right? During the uridial stage. When it start producing teleal stage, it will become black, right? So usually now you are seeing it as a uridial stage. Once it is getting converted into teleal, means it will be becoming black in nature, right? Then this is the size of it. Then what is the predisposing rainfall, right? Then low temperature, right? Is it audible, right? Please confirm me once. Is it audible?
Yes, sir. Yes, sir.
So the temperature is the optimum temperature is 20 to 25 degrees Celsius. Okay, so we can usually, instead of telling the concept of optimum, we can stick on to the cardinal temperature. Okay, so cardinal means it will start from it will encompass low, medium and maximum. Okay, or low, medium, or it can be optimum. Okay, so for a pathogen, the 3 requirement is needed, then only we can tell or we can justify the behavior of the pathogen with respect to disease. Okay, so optimum should be there anyway, that is a mere estimation, but if you have lower and maximum temperature, then we can check a trend of the particular pathogen, how it is following, whether it is going in a normal distribution or it is having a trend like this. Okay. So that's why we, if it encompasses the studying of both lower optimum and maximum. Okay. So these will compass as a cardinal temperature. Okay. So the optimum is usually 20 to 25 degrees Celsius, right? Then the severely infected leaves turn necrotic and desiccate, but are attached to the plant. Okay. Then the disease is usually found when the plants are about 6 weeks old. Okay. At later stages of the, or in adult plant, we could be able to see this infection. Okay. So see, in severe infection, lower leaves dry and droop prematurely, which also leads to production of small shriveled leaves. Right. Here you can see how the rust infected field looking like, right? Okay. Then coming to Alternaria leaf spot, which is caused by Alternaria arachides, right? So here, you know that usually Alternaria will cause target board symptom, right? That is nothing but it will look like a target board symptom or different circles it will be having, right? Like this. But in case of groundnut, we can't see this distinguishable character, right? So just like any other kind of leaf spot or blight, it will be causing a infection, right? More prominently. So here we may not be able to distinguish proper target board symptom, but usually as the leaf spots, it causes like lesion or necrosis followed by a yellowing. So those things can be seen, and usually it will start from the lamina. Okay, or sorry, marginal lamina. Okay. So this is about the Alternaria arachides. Then stem rot or sclerotium wilt caused by Sclerotium ralsei, right? So to identify this disease, first what you can do, it can come to a over range of starting from seedling to adult stages. Okay, even at the pod bearing stage also. Okay, so previously it was not showing in pod bearing stage also, right? So what you can see, whenever if the seedling death has occurred means, right? So you can check for the production of mass mycelia here, right? So here you can check for the mycelia whether present or not. That is one thing. Then you can check for the sclerotial bodies. See here how the sclerotial bodies are there, just like a mustard seed. So you can check for that mustard seeds like asexual fruiting bodies or asexual sclerotial bodies, whether find on the ground or not, right? That is one thing. Then followed by, of course, I told you, right? It will be drying, how the wilting of the physarum will occur, right? The same way at the seedling stage, or the how the damping of occurs, right? The same way it will make the seedling to topple over, right? That is one thing. In adult plants, how you can just know means, just you can pull out the dyed one and you can check for the white mycelial growth throughout the stem or the root, right? Then you can easily find. And even on the pods also you could able to see the white fungal growth, right? So this is the criteria how you can identify a Sclerotium ralsei, right? So basic criteria you can check for the, easily you will find once you enter the field, you can easily check the white mycelial growth in the stem or the collar region, right? So again, if you want, this is the major conformational, because you may be creating confused with whether it is a rhizoctonial sclerotia or Sclerotium sclerotia. Okay, so that's why this is the basic criteria you can check. Then again you can stick on to this one, then you can confirm whether it is a Sclerotium or not. Then dry root rot, right, which is caused by Macrophamina phaseolina and Rhizoctonia bataticola, right? So it also causes the same thing, right? But more often it will come on the adult plants. There it was coming on both seedling and even the adult, right? But here it is more often of adult plants, right? And even in case of Sclerotium, it is more of wherever the saturation is high, then you can check the, you can see the sclerotium infection because it requires high moisture content in the soil. Okay. But in case of dry root rot, as the name only indicates, it requires very dry. Okay. So in semi-arid conditions, usually the infection of this dry root rot will be very high, right? Takes place, right? Then followed by, once you just pull out the plant and you can check the root, it will be striated. Okay. Usually the skin will be peeled off. Okay. The skin can be easily peeled off just if you remove the root skin. Okay. So that is the one criteria to identify, and the blackening of the roots will be there, right? And even the plant will completely dry. It will be like black or brown. But in case of sclerotium, usually you will see a sudden death or even a yellowing or normal greening wilting will be there, right? But here usually it will keep on drying and it will show black to brown color plants. Okay, so these are all the basic criteria how you can identify, and even here also you can able to see the sclerotia. Okay, that will be irregular in shape. There it was very regular, like it was mustard seeds like shape, but here it is a irregular. Okay, so this is how you can differentiate, and it requires low moisture content. Okay, when there is a moisture stress in the field, the infection will be very high. Okay, the dry root rot will take an advantage of high moisture stress, but in case of sclerotium, it will take an advantage of low moisture stress or high moisture, right? So this is the difference between dry root rot and sclerotium, right? Okay. Okay, so peanut stem and bud necrosis, right? So already I have told you, right? So stem necrosis caused by Hilar virus, right? Bud necrosis caused by tospovirus or orthotospovirus, right? So both this is pollen transmitted, right? This is the trips transmitted. So as the name indicates, usually the bud necrosis, the buds will be deteriorated, and here on the stem it will cause a oak leaf symptom. Okay, or even the leaf also. Okay, so the root knot nematode, Melodogyne, right? These are all the different species, right? Alternaria, Hapla, and Javanica. Okay, so usually it will cause knotting on the roots, right? So you must be knowing. So what are all the symptom usually a nematode cause, right? So starting from, it is similar to the viral infection what it does, right? So starting from yellowing, right? Then stunting, right? So it is also a kind of obligate in nature, right? So it requires the host to be survived if it wants to survive, right? So if that is the case, it will try to not to kill the plant, right, by extracting its nutrient. So usually it will make an knots and it will survive, right? Okay, so coming to integrated disease management, right? So we have studied the disease of groundnut. So what are all the idiom practices we can follow to check the disease infection or the disease, right? So you should start from the basic to advanced. Okay, you should not, it should be like that. Okay, so because the whatever the BC ratio you can have, that should be followed. So the cultural method, right? Removal of voluntary plants, because you know that the rust of the groundnut will be to do remove, they will be harboring their plants because not volunteer. So you have to take care about the rogging out of voluntary plants, right? Then crop rotation. So non-host crop should be planted, right? Because you know that even like so many peanut clump virus, right? So if cereals are there, means its infection will be very high. right? So you should take care which are all the non-host crop for this particular disease. So like that, you should know what are all the non-host, or instead of knowing non-host, you should know the what are all the host range. If you take out the sclerotium means, right, it is having the highest host range, especially in case of pathogens, right? It is having highest host range. So you should take care whether, because if you check, it will come on chickpea also, sunflower also, right? Even most of the pulses also, and even oil seeds. So there is a broad range of even rice, right? So many host it is having, right? So you should check out whether it is a non-host or not, like that. Then keep out the weeds, right? Then the time of sowing and spacing should be well planned. So this is also most important criteria. Then field sanitation with respect to any disease, you should have to control that. You should have your field should be sanitized. Then deep summer plowing is helpful because you know that sclerotial bodies, which are very resistant structures, right? They will be harboring inside the soil, right? So to avoid that, what you have to do, you should do summer plowing, right? So minimum 30 centimeter, the plow should be drawn and it should expose to the sun, right? For up to at least 2 to 3 months, right? Then the temperature will be at least raised to 40 to 45 degrees Celsius, so that the whatever the sclerotial bodies are, the mycelial bodies are there, it will get affected, right? Then the regulatory methods, strict plant quarantine regulation should be enforced, right? Because I told, see in groundnut, like aflatoxin, right? Because its toxicity, it should be, it is regulated, right? The same way you can see peanut stripe virus, right? It is also seed transmitted, so it should be regulated with a thorough quarantine procedure, right? So like that, use of certified seed should be there. Then biological control, whatever may be, it should be followed with the Trichoderma viridae or Arzianum, right? So whatever the or Coenji, so etc. Whatever the species available, which is showing very effective, right? So it should be treated with the seeds, right? Then it should be sown, and even Pseudomonas, right? Or even Pasturia, for example, against the nematode, right? So it should be treated and it should be sown. Okay. Then chemical control, carbendazim or thiram. So thiram usually we will be going for the seed treatment, right? Even carbendazim can also be seed treated, right? With respect to, like say, collar rot, right? Or even the whatever you can call it as sclerotium rot. Okay, so sclerotium rot, you should be applying the Vitavax power, right? That is nothing but thiram plus carboxin plus thiram. Okay, so that will give an effective control against the collar rot, even the sclerotium wilt, right? Then the spraying of the respective carbendazim or mancozeb or even hexaconazole, right? Or even the propiconazole, right? Against the rust, it can be used at 0.1%, right? But for leaf spots, even combination of propiconazole with the dithiocarbamate is also possible, right? That also gives a good advantage. But even most preferred, especially in southern Karnataka and even North Karnataka, it will be like Kavach, right? So when it comes to late or early leaf spot, they will go for the spraying of Kavach, right? Chlorothalonil, right? So at 0.2%, so it gives an effective control, right? Okay. Do you have any doubt with respect to groundnut diseases? If you have any doubt, you just ask.
Dear participants, please do interact with the Vimal sir. It will help you. If you just keep muted yourself, it will not be that much helpful. So please be interactive.
Okay, then shall I start with the next disease? Okay. Sorry, crop. Sesame, right? So coming to Alternaria leaf spot, right? So it is caused by Alternaria sesame. Right? It is not visible. Alternaria system, right? So this is the causal organism. And as I told earlier, the Alternaria means usually it will cause the target board symptom, right? So here you can see what is the target board symptom, right? Here you can see exactly how the target on the leaves, the target board symptoms are there, right? Okay, so usually on the leaf it will be having encircled spots. Okay, so this is the characteristic, right? And if the infestation or infection is high, means it will not see whether it's a leaf or a pod. Okay, so throughout the plant except root, the infection will be there. Here just you can see on the pods also how much infestation, and even the pedicel and even the stem also, right? So infection will be very high. Okay, and of course anyway you know that it is not an obligatory, so it will just droop of the leaves, and even the plant yield will be reduced very drastically. Okay, when if the infection is high means. Okay, so here you can see the culture how it will be looking like, and it is a more of late pathogen. Okay, so if you take the other pathogens, it will easily come within, say you can 7 to 14 days, the plate will be covered. Okay, if you just take a sclerotium and even the aspergillus, within 7 to 8 days the entire plate will be about 9 centimeter will be covered. But if you take the Alternaria, it will take nearly 21 months to grow even 3/4 of this plate. Okay, so that's why it is usually late pathogen. Okay, and even the structure you can see, right? Usually it's a colored, okay, colored pathogen, right? Colored conidia, and as the conidia shows here, it is having muriform conidia, right? That means it will have both transverse and horizontal. Okay, so both septa will be there, right? So, and it will have a beak. That is the important character. Then coming to next disease, that is powdery mildew. So it is caused by either oidium or Spirotheca phylogenia or even Levalula. Okay, so reports are there where even different pathogens infection was seen on this particular crop. Okay, so as you know that usually if it is a oidium and Spirotheca, right? If it is a oidium and Spirotheca, it will be more on the upper leaf. Okay, here it will be there more on the upper leaf, right? But if it is Levalula, it will be more on the lower surface. Okay, so it is more on the lower surface. So that is the more important criteria to differentiate between these two. Okay, so if it is a Levalula, right? See here you can see it will produce the candle shaped conidia. That is the primary criteria. Then it will have a branched conidiophore, right? See here it is a branching, right? But in oidium you may not be able to see those things. Just it will be cylindrical, one by one it will be keep on bearing conidia, right? But here you can see it is a branched one. Okay, so the conidia will be candle shape in nature. Okay, so these are all the two important characteristics when it comes to Levalula tarica, right? So, and this is the Levalula is nothing but perfect stage, right? It is a semi-endophytic like this. It will bear the myceloid appendages with a mini asci inside the clistothecium. Its asexual stage is this one, right? Okay, so the next disease is phyllodes. Okay, so as you know that it is caused by a phytoplasma and it is transmitted by albicinctus, right? So Orochius albicinctus. So in last class only I have described symptoms and everything about this, right? Then coming to charcoal rot, which is caused by Macrophomina phaseolina. So as the name indicates, charcoal rot, it is mainly with respect to the stem, right? So the same criteria what I told in groundnut, same applies here also, right? The the most important character what you can see means striation, right? The stem will be striations will be there or striped. Okay, so you can easily distinguish between individual pith region or thread-like structures will be formed. The entire stem will be disintegrated into thread-like structure. Okay, so that is called. So that is the most important characteristic with respect to charcoal rot. And even if you just check out the stems closely, right, you can see the sclerotial bodies attaching on the stem, right? So that is the one criteria. Then just as I told, the root skin of the skin will be easily peeled out. Okay, so that is of the one criteria, right? Just if you just check on the media, darkish black color mycelia can be seen. Okay. Then coming to Sarcospora leaf spot, which is caused by Sarcospora sesame. So usually it will cause more of frog eye symptom, right? So leaf will be there. The center will be of gray color and surrounding will be brownish, right? So it will be kind of aesthetic look it will be showing, right? So you can easily identify between Alternaria and even the Sarcospora. Okay, so Alternaria will have a target board symptom. It will be of more of brownish color, but here you can see grayish centers, right? So this is the one criteria where you can differentiate between Sarcospora and Alternaria, right? And once infection will be very high, the blighting will take place. And this is bacterial leaf spot, which is caused by Pseudomonas syringae sesame, right? I think this one also I had covered in the last class. Okay. So how you can effectively manage diseases in the sesamum, right? Right? That's a disease, right? Okay, as per of your crop also, right? So these are all precautionary measures what you can do, right? Then ultimately we can go for the chemical, right? Especially in broad range, carbendazim spray will be, it will be sprayed most preferably. Okay, if it is a Alternaria infestation is high, then we can go for mancozeb. Okay, because why still mancozeb is ruling means it is a contact fungicide. That is the one reason. That's it. Okay, but these are all whatever we are recommending. It is all of systemic, right? So that's why it will keep on changing. Then we can have even hexaconazole against the leaf spot. Okay, so powdery mildew and leaf spots, colorless conidias leaf spots can be managed through the carbendazim or propiconazole or even the tebuconazole, right? Okay, then coming to castor, right? So castor wilt, which is caused by Fusarium oxysporum pharma specialis resini, right? Fusarium oxysporum pharma specialis resini. So how I can identify whether a castor plant is infected with the Fusarium or not, right? So the first criteria what you should do means first you check whether partial wilting is taking place or not, right? So partial wilting means some parts of the plant will be healthy or greenish. See here you can see how the some parts of the plant are very green, right? Or here you can see exactly, see it is standing, but here the some part of the branches are getting dry. That is called partial wilting. Why it happens? Because it will start infecting the lateral roots rather than the main root, okay, or axial root. So that's why when the lateral roots are getting infected, so it will be the food supply will be partially available. So wherever it is not available, their wilting will be taking place due to the blockage of the vascular bundles, right? So that is the reason behind the partial wilting. Okay, then yellowing will be there, right? So yellowing you can see in the plant, that is also another criteria, right? Then followed by, you can just break open the stem and you can check blackening of the middle region. That is called vascular bundle region should be blackened, right? So that is also one of the criteria to identify this. And if it is early means, okay, so the entire branch may or branch or even the leaf can be drooped off, okay. Okay, so these are all the criteria what you can find with respect to Physaria, right? Okay. Then coming to rust, right? So which is caused by Melonspora ricini, right? So just have a note that most of the rust disease, you just leave cereals, okay? So because cereals, irrespective of surface of the plant or the leaf, it usually occurs, okay? But in case of other crops, if you take, usually the rust will try to concentrate on the lower surface. Okay, exceptions are there like chickpea and other things. But if you take a generalized statement, most of the rust will be occurring on the lower surface where you can identify, right? So here you can see usually it will be yellow to orange color, then it will turn on to brown to blackish color whenever it starts producing the pterospore, right? So this is the identification criteria. So the underside of the leaf will be producing the pustules, right? It is nothing but the epidermis will be broken out and the pathogens will be coming out of the cell by breaking this epidermis. Okay, that's why the spores will be, or the urinal spores will be accumulated in pustules, right? So once it aging takes place, it will turn into teleospore, right? So the corresponding surface of this particular leaf will be having, right? The corresponding surface of this particular leaf will be having yellowing, right? So as the senescence starts, the drooping of the leaf spill takes place and it will also cause the drastic yield reduction, right? So it is caused by Melonspora ricini, right? So it is a single cell. Okay, so see, usually the Melonspora will be like this in palisade manner, it will be arranged. Okay, this is the teleospore of the Melonspora. Then Sarcospora leaf spot, which is caused by Sarcospora ricinella. Okay, so here also, instead of, it can be gray to light cream color, right? So this is the center of the leaf spot, right? Then the outer margin will be slight brownish in color, right? And again the outer of that will be having yellowing, right? So these are all the criteria or how you can differentiate between Sarcospora leaf spot. And once the infection progress highly, then the blighting will take place, right? So here you can see the individual spots are there. It is getting blighted and means coalescence will be there. Then it will coalesce and it will become blightening. So when it blightening will be there, usually it will just tear off the lamina. Okay. Then Alternaria, that is Alternaria Blight, you can call. It is caused by Alternaria ricini. Okay. So the same here, you may not be able to find this kind of gray or cream color centers, right? So usually it will be more of brown in nature, or it will turn into blackish, right? Then overall blightening will be there by turning down the lamina. Okay, so at higher infestation, the lamina will be collapsed. Then the next disease, that is charcoal rot, right? Macrophomina phagorina. This is also major disease, okay, as far as I have seen with respect to castor. So usually the same symptoms what it does with respect to other crops, right? See, I told you, right? The peeling of the skin will be the major symptom to identify this, right? The peeling of the skin is taking place, right? Then followed by the blackened mass will be there. That is nothing but mycelia, right? Because the thing is that because of the dryness of the plant, that due to this infection, the peeling of the skin of the root will be there, right? Then blackening of the mycelia will take place. Then the whatever the stem is there, right? So it will be striated, right? The pith will be disintegrated into a thread-like structure, right? Then the entire plant will start drooping. Okay, so here you can see the wilting. Okay, so if you want to take down the life cycle, means see here you can see inoculum in the soil, right? Then it will start germinating. Microsclerotia will be there, right? And as I told, at high moisture stress, it will be there. That means the temperature will be very high at the soil. See, it is about 30 to 35 degrees Celsius, right? Then the germ tube production will be there, right? Once it will get the stimulation, then it will start producing the germ tube. Then again, it will just differentiate into aprosoria. Then again, penetration peg, right? Then it will start invading the tissue, right? Once it has invaded, then it will try to recognize the particular recognition molecules. Okay, whatever the receptor and ligand is present, then it will try to recognize the particular host, whether I am able to cause the disease in this particular host or not, right? Then once it is done, then it will try to do invasion and colonization, right? So it will sophisticatedly, it will just sit on that and it will thrive. Then once the life cycle completes, then it will start what you can tell, spreading of the particular propagules after its reproduction. So like this, the cycle will continue. Okay. Then gray mold, gray rot, or blossom blight. Okay, so these are all the different terminologies we have adopted with respect to Botrytis ricini disease, right? So that is gray mold, right? Gray rot, or even the blossom blight. Okay, usually examiners will be keep on confusing this the way they will ask the question with respect to the students. Okay, so you just know whether it's a gray mold, gray rot, or blossom blight. So all are same only. It is caused by Botrytis ricini or Botrytinia ricini. Okay. So as the name indicates, the symptoms can be a kind of syndromes, what you can call. So it will start from either it can cause infection on leaf or fruits, right? Or pods or twigs, right? So, and even the leaf. So everywhere except root, it can cause the infection. So on the pods also the infection will be there. That means usually at high moisture content, whenever the high rainfall is there, the infection will be predisposed, right? So that's why there will be huge rotting will be there, right? The black colored or gray colored mycelia will be clumped up on the particular inflorescence, right? So here you can see how, and if you just keenly observe either under microscope or even any lens, you can see the branching of the particular mycelial and its spores. Okay, so Botrytis, you know that it is a scientific name of cabbage or cauliflower, right? So that's why the name Botrytis. So that's why it is like looking like a cauliflower head. Okay, how the flower will be, right? The same way the spores will be arranged. Okay, so that's why if you just keenly observe, it is of a single celled only, but it will be very clumped so that it will be looking like a cauliflower head. Okay, so that's how the spores will be looking like. Even under skin, lens also you can see. Then the one more differentiating criteria is on the inflorescence, you can see the blue colored lesions. Okay, the blue colored patches will be there, right? So that is the most important criteria to identify. And here you can see how the entire mycelia has covered the pods, right? So like this, it will be covering entire the pods or the fruits, and after that the oozing will be there. Okay, so the gummosis will be there from the infected, what you can call it as inflorescence. Okay, that is one thing. The drying of the leaves will take place. So these are all the, and even the sometimes lesions can also be seen on the leaf. Okay, so these are all the kind of symptom it is caused by botrytis. Okay, then bacterial leaf spot, right? I think last class also I covered this. So it is caused by Xanthomonas campestris racini. Okay, so usually it will cause water soaked lesion, right? Again, it will cause spots. Okay, then seedling blight, which is caused by Phytophthora parasitica. So as the name indicates, seedling blight, right? It is mostly confined to the seedling, right? So at early stages of the crop, what it does means it will cause the stem necrosis, right? That is the one criteria. And you know that usually Phytophthora is preferred at high moisture content, right? So when high moisture is there means it will just cause infection, right? So it will cause blightening of the stem, right? Once blightening takes place, then the entire plant is getting infected, right? So the leaf will be curled down and the blackening of the leaf will take place. Okay, so the Phytophthora parasitica is the causal organism. Okay, so how to manage castor, right? So as you know that it's oilseed crop. So usually you have to go for the proper cultural practices. Like say, as you know that you should give an weightage to a particular disease or a group of diseases or even a hierarchy, right? Which one is having more, like say wilt, right? Or rust, like that you can categorize which one is having like that. Then accordingly, you should plan your management strategies. Okay, the first thing, foremost thing, whatever may be, you should follow the sanitation. It is minor or major. So the field should be sanitized first, right? Then you should follow the cultural practice, like you should avoid the volunteer plants. That is one important criteria, right? Then whatever the weeds are there, you should rogue out, right? Then yeah, summer plowing, because the how much important is this summer plowing means, you know that whatever the soil borne pathogens, that is the major criteria, right? Because you don't have the fungicides and any other method to control the fungal soil borne pathogen, right? So the only criteria what you can do for is, like you can go for addition of amendments, right? Or you can go for addition of doing summer plowing, right? So these are all, or you can go for the rotation of the crop. So these are all the basic criteria what you can follow, and even the resistant varieties, right? So even especially in case of this, the fungicide is merely effective. Okay, especially in the two wilt diseases, it is Fusarium wilt, it is very doubtful because it is a sporadic in nature, right? If you have sclerotium means, okay, somewhat we can control with the drenching, but if it is a Fusarium wilt is very difficult, because here and there, if it is a field means here and here and here, it will be infected. Okay, so when the irrigation is there, like whatever the furrow irrigation, whatever you are following, so the pathogens will be keep on traveling. So if that is the case, it will start spreading with respect to each neighboring plant. Okay, so in that stage, it will be very difficult to control. That's why, so we should take care of the precautions rather than when it comes, right? Then after cultural, okay, whatever the biological is there, you can follow, right? Like trichoderma, then pseudomonas, right? So these are all preferred. Then ultimately, if you want to go for chemical control means the same way, the Fusarium, it can be controlled with the carbendazim, right? So Binomil, I don't think it is most probably it is available, but some part it is available also. Okay, Benomil, right? Then even triazoles compound you can follow, right? For spray like Cercospora and Alternaria, right? So those things and even the rust, it can be controlled with the spraying, right? The same chemicals what I emphasized on the sesame, right? Or even the groundnut. The same things you can follow here also. Then coming to next disease. Any doubt? If you have any doubt, just ask me. Okay. Then coming to next crop, that is sunflower. So downy mildew, right? So which is caused by Plasmopara allsteady. So as you know that it is a oomycetes fungi, right? So the initial symptoms will be at even seedling stage, the infection can start. If it is infecting at the seedling stage, there will be drastic stunting will be there. Okay. Just like how the viral disease will cause now, the same way, if it comes at early stage, the stunting will be there and the plant will unable to stand up and the complete yield reduction will be there. Okay. Because it will not produce any reproductive organs. Right. So that is one criteria. Then that downy growth will be there. Okay, see here you can see the downy growth under surface of the leaf, right? Above surface will be corresponding yellowing or chlorosis will be there, right? Yellowing or chlorosis will be there above surface and downy growth will be there underside of the leaf. Okay, so here also you can see how the downy growth, and of course the head will be erect. That is also one of the key important criteria whether it is having viral infection or fungal infection. To differentiate, the heads will be very erect. Okay, so that is the one of the identifying criteria. Okay, so I think these are all more than enough to identify whether it is infected with the downy mildew or not, right? So it is caused by Plasmopara allsteady. So you know that the Plasmopara is mycelium branching in a 90-degree angle, right? So like this, the sporangium will be branching at the 90-degree angle, right? So it will be inside this Plasmopara, right? It will have so many, what you can call it as, yeah, sporangium will be there. That's it. Okay. Then head rot, which is caused by the Rhizopus species, right? So there are so numerous species which are coming and infecting the sunflower. So here it is a majorly a secondary pathogen. Okay. Why we will call it a secondary pathogen means first it fails to infect the host directly. Okay, so healthy sunflower is there, right? If I just leave a Rhizopus spore on this, it fails directly. Okay, or oftenly it fails. I should not tell every time. So oftenly it will fails to get established inside that because of its toughness of the cell wall or whatever you can call it as. But once the insect infestation is there, see here, once the insect infestation is high means, as you know that the insect can easily break down the whatever the cell component is there and it will expose the cytosol to the particular pathogen, right? So then what it does means easily the fungus can enter and it can colonize the particular cell or tissue or host. That's why we call it as a secondary pathogen. Okay, so when insect activity is high, right, then the Rhizopus activity will be very high. So this is the correlation, right? So if you want to check out the disease infection also, you can get a good correlation between the insect activity and even the Rhizopus infection. Okay. So this is how you can check. And the head rot will be, as the name indicates, behind the what you can call head, right? The soft rot symptoms will be there. Rotting will be there. That rotting will be covered with the black colored mycelia. It is nothing but Rhizopus mycelia. Okay, so full blackened mycelia will be there. And if you just keenly observe either under the microscope or even the lens, you can see this is the sporangium and it will start bearing the Rhizopus sporangium. Inside that, lot of aplanospores will be there. Okay, so this is what the Rhizopus will look like, right? So as you know that it will have stolons and the rhizoids, right? So this is how the rhizopus will be looking like, and it is a secondary pathogen. Then Alternaria blight, right? Alternaria helianthe, the same, just like in other symptoms what you have seen, but here is of more blightening nature. Okay, so sometimes you can see the target board symptom also. It's not an it's an obvious, right? Then if you just check out, the entire blightening will be there on the leaf. If you just take out the leaf surface of this sunflower, means it will be high, right? In case, especially in case of hybrids, it will be broad. So entire leaf lamina will be covered, okay, not leaving even 1/4 of the area. So like that much area will be covered when the infection will be very high. Okay, so that means blightening will be there rather than spots, right? So once the blightening will keep on increasing, you can just see here how the drooping is taking place, right? So this much drooping will be there, and entire yield, whatever the seed quality will be reduced, right? The oil production or oil content of the seed will be reduced, right? So the plant performance will be very poor, right? So even you can see, as I told in earlier also, not only on lamina, right? Even you can see on the pedicel, right? On even the stem and even the head also. So this is the importance, and even you can see the sepals, right? So on these also you can easily able to see. And even the next, coming to powdery mildew, which is caused by Erisife chicoracearum, right? So yeah, so if you just check out, it is causing mainly on the upper surface. Okay, here we are able to see the Erisife, but if you just check out the other countries, like especially in USA and those things, they are coming across with the Golovinomyces. Okay, this is a earlier synonym. Okay, but this is a now close relative of Erisife only. Okay, so this is another genus. Usually it is not observed in India, but in other parts of the country we could able to see the Golovinomyces chicoracearum, right, which is causing powdery mildew on the sunflower. So the same here you can see, in case of downy mildew, the underside surface of the leaf was having downy growth, but here it is having upper surface is having high powdery growth. Okay, so usually it will be powdery in nature, right? Then the plant, usually if it is early stage, means stunted, okay, the of course the yield will be reduced, because the whatever the photosynthesis area is there, that is covered by the particular mycelia, right? So it will not allow the effective or efficient photosynthesis, right? Okay, so the next disease is rust, which is caused by Puccinia helianthe. See, initially it is also mainly preferring the lower surface, right? So it will be mainly preferring on the lower surface. Initially it will be orange to brown, so you can take it as a brown. Okay, then once the T-lel production keep on producing, then it will turn into black color. Okay, so after that, if you want to identify whether it's a rust or not, you can just rub. See, here you can see the rust content on the fingertips, right? So this is how you can easily identify whether it's a rust or not, right? So it will just break down the epidermis, and it will come out and it will expose its pustules, right? So if you just check out the disease cycle of the Puccinia, right? So you can start from basidospore, right? So basidospore single celled, right? N. So it will just fall on the receptive hypha, right? So here you see it is a autoecious, okay, because anyway, see, this is the sunflower. So now you have cultivated, sown, and here on this the infection will take place. So after completion of partial life cycle, it will go and fall on the other plant or volunteer plants, or you can consider this one as a volunteer plant, right? And it will go, the rest of the new cycles will be completed on the new sown crop. So like that, it will complete the life cycle. That's why it's an autoecious. Okay, so it does not require another host. It requires another host in with respect to same category host, right? Sunflower only. We are not, if it should be called as an alternate host, means it should be of another family, right? But here it is of a same species, right? That's it. So basidospores will fall on the sunflower. Again, it will combine with the receptive hypha, right? Sorry, it will produce the, whatever you can call it, a pycnium, right? So once the pycnium is produced, in two different stages, it will produce pycnospore and receptive hypha. Okay, it might be on the same leaf or it might be on the different leaf. So once the compatible, either you can call it as pycnospore and receptive hypha comes, then it will combine and it will produce the acium, right? It will produce the, okay, so it will produce the acium under the surface of the leaf. So once the acium is produced, right, then it will keep on producing. See, aciospore is nothing but n plus n in nature, right? So here a karyogamia has not taken place yet, so it will become n plus n, right? Here it will be n only. Once the receptive hypha and pycnospore combine, the n will become n plus n. Then from here after it, the n plus n condition will be carried, right? So again, it will get into iridium, right? Then iridium will produce lot of iridospores. It is also condition of n plus n, but the n plus n, you know that it is of repetitive nature. That means it does not require the other stages to complete. Just the n plus n condition go and again it can produce another n plus 1, right? So that's why it is called a repetitive spores. So because of this only, there will be a lot of epidemic instances, right? So once the iridospore is produced, then it will stick on to the karyogamy. That means the nucleus of the both the spore will be combined, right? So this is the one spore. It will have one nucleus and another nucleus. So these both will be combined and it will become 2n, right? So that is called, it will produce teleospore. So teleospore, it is nothing but it is a stalked one, right? Like this it will be there, and it is a usually bicelled, right? So yeah, so like this it will be there. And here you can see once the teleospore is there, then it will produce the promycelium. Then on the promycelium, it will produce the basidiospore, right? Sorry, basidium will be there. Then upon that, basidiospores will be there. So once the basidiospores will come, it will produce the primary mycelium and it will start producing the respective pycnium, you can call it as. So this is how the life cycle of the particular Puccinia will take place, right? So the next disease is basal rot, which is caused by Sclerotium rolsi. So already I told you in groundnut only, the same symptoms here also applies. So if you want to identify, just enter into the field, check on the stem or the collar region whether white mycelial growth are present or not. Then again, if you want to confirm, means just you check whether mustard seed like, right? Mustard seed like what you can call sclerotia, right? So sclerotial balls are present or not. That should be noted down. Then of course the wilting will be there and stunting will be there, right? Okay. Do you have any doubt? Okay. Then safflower, right? So the safflower, which is with respect to Alternaria blight, which is caused by Alternaria carthami, right? So here also you can see target board symptom. I assume you could able to see here, right? See, lesion is there inside that. A target board symptom will be there. So this is of the blight, and it will, once the infection is high, then it will coalesce and it will entire lamina will be brown, right? And even sometimes you can see the blotching type symptom with respect to Alternaria. Okay. Then, so I told you causal organism, right? Alternaria cartham. Then coming to wilt, right? Physiarium wilt, that is Physiarium oxysporum forma specialis carthami, right? So here most of the diseases, the species are carthami only with respect to a disease of safflower. Okay, so just you can have a rough guess that whether if it is a rust, means it must be either Puccinia carthami or not, like that. Okay, so most of the times it is generalized that it is a species of carthami. Okay, so the symptoms are same, right? So as usual, partial wilting, right? Then plugging of vascular bundle, right? Then you can check out the lateral roots infection initially, then it will turn on to taproot also, that is called complete wilting. right? Then you can see yellowing, and anyway the yield will be drastically reduced, right? So this is how you can differentiate. Then rest, see, Puccinia, calcitrape were centuriare, right? The synonym also you can call it as Puccinia carthami. Okay, so many synonyms are there. So one more is Puccinia, calcitrape were centuriae, right? So here also the same. Usually it will infect the underside of the leaf, right? And the iridospores will be transformed into the heliospore. That will be looking like a black colored pustules, right? Then after that, at senescence stage, there will be high defoliation, right? Defoliation will be high. Then powdery mildew, right? Spirotheca, Phylogenia, right? So here also the mostly the infection will start both on upper surface and even the lower surface, right? So the infection will be there, and the powdery growth can be seen, right? So that is the basic criteria. Then followed by anyway the seed or the yield reduction will be there, and even the corresponding leaf area will be yellowed. That is also one of the criteria to identify, right? So these are all enough, I think, to identify whether it's a powdery mildew or not. Then Spirotheca, it is similar of Erisife nature, right? Like this, the Clistothecia will be there with a only single assay. Okay, inside the single assay, 8 ascospores will be there. So like this, it will be looking like, and it is a ectophytic in nature, Spirotheca Phylogeny, right? Then the next crop is mustard. So the disease is caused by, downy mildew, right? So the disease is caused by Pernospora parasitica, right? So the disease is caused by Pernospora parasitica. So what you can see means the under surface of the leaf, you can see the downy growth. Okay, so that is the major criteria, and deformation of the leaf will be there. Okay, the structural integrity of the particular leaf will be deformed. Okay, so that also one of the important criteria. Then of course rest of the thing the same, the yellowing will be there, corresponding leaf area will be yellowing, right? That is also one of the criteria, and stunting will be there at initial stages. Then so Pernospora, as you know that it is a homicide fungi, the acute angle branching of the spongiophore will be there, and at the pointed tip it will bear the round shaped sporangia, right? And it will not produce the zoospores, it will germinate through the direct, or it is having a direct germination. That means the sporangium will directly produce the germ tube rather than going for the zoospore production, right? Then white rust, Albugo, Candida, right? So if you want to identify whether it is a white rust or not, just turn down the leaf, and you can see just like how the whitefly will lay the eggs, the same way the Albugo Candida will be there. Okay, from far away. Just if you just take out the keen observation means you can see the raised portion of white colored masses. Okay, rust. That is nothing but white colored rust will be there. So like that you can easily identify, that is one criteria, and the corresponding area will be yellowed. Okay, so that is the one criteria to identify, and it will cause Sag Head Symptom. So Sag Head Symptom is nothing but whatever the reproductive organs are there, right? So that will be transformed into how the deer's head, right, or the horn will be there. The same way it will be there. Okay, so like this, whatever you can call it as, so it will be, the head will be like that, so it will be very twisted, and inside that the sporangium or the oospores, whatever may be there, so it will be filled up. Okay, so you can able to see in both the downy mildew and even the white rust. Sag Head Symptom. So this is called systemic infection. Okay, systemic and localized infection. Localized infection. Okay, so this is localized infection, this is systemic infection. Okay, then Alternaria, right? Alternaria blight, you can see Alternaria brassicae or Alternaria brassicola, right? So I think here you can able to see the exact target board symptom, right? See here how much accurately it has targeted it, right? So like this. Okay, so this is the one of the criteria to identify whether it's a Alternaria or not. So usually the brassicae will be usually of light brown color. Okay, the Alternaria brassicae. If it is a brassicola, right, it will be of more black color. So how to identify means cola, right? So like Coca-Cola, it will be very black. So like that you can remember. So cola means it will be of high blackness, so that you can merely differentiate whether it is a what kind of species the infection was has taken place. Okay, so like that. So initially it will be spots will be there, then the blightening will be high, right? So this one Alternaria brassicola. Okay. Okay, do you have any doubt? If you have any doubt, just ask me. Okay, so here we have completed the DS of oil seeds. If you have any doubt, please ask. Or shall we have question session first? Okay, so just answer me the question. The statement 1 says the rust of sunflower will have flecking on the leaf before teleospore production. And statement 2 says sunflower rust initial phase completes on sunflower itself. What is the answer? Please answer me. You should answer. Or you have understood everything, or you did not understand even a single point. So that will be the conclusion if you are not answering. Please answer first. Which is the answer? The statement 1 says rust of sunflower will have flecking on leaf before teleospore production, and statement 2 says sunflower rust initial phase completes on sunflower itself. I think it is not an harm to guess also. You can make any guess, no issues. Is it audible? Answer me, anyone. Is it audible? Is it audible, right?
Yes, you are audible.
Okay, okay.
Students, please interact whenever sir asks something. Hello.
Okay, so see, the answer is, as I told, see, the rust of sunflower will have flecking on the leaf. Of course, it will have flecking. Flecking is nothing but kind of chlorosis. Once the chlorosis occurs, then only the infection will takes place. Okay, so it will have flecking right before the teleospore production, then the pustules will be there, right? So this first statement is correct. As I told, the sunflower rust initial phase completes on the sunflower itself, right? So this is also correct statement because it doesn't require any other alternative host or alternate host, right? So it completes its autoecious nature in the one freshly sown crop and even the voluntary plants, right? So that's how the both statement will become correct. Can anyone answer this? Melamspora teleospora is having dash shelled. One, two, three, or many? Okay, it is having one cell. Okay. Then statement 3, question number 3, statement 1. Mancozeb is effective on Cercospora than carbendazim, right? Here it is asking about the Cercospora, and statement 2 says stag head infection can be seen both from downy mildew and white rust, right? So the first one is wrong because it's a carbendazim can have high effective control rather than mancozeb, right? So in generalized statement, what you can have means mancozeb will have more control ability on colored conidias, and the colorless conidias will have more affinity towards the what is you can call it as susceptibility on carbendazim. Okay, so like that you can assume. So the first statement will become wrong, and the stag head infection will be we can see on both downy mildew and even the white rust. So the statement 1 is wrong and statement 2 is correct. That means statement 2 is correct and whereas statement 1 is wrong. Okay. Yeah. Then question number 4 says blue spots can be seen on which of the following diseases? It's color rot of groundnut or gray rot of castor or white rust of mustard or downy mildew of mustard. Okay. So here the gray rot of castor, I told you, right? The botrytis can able to cause the blue spots on the inflorescence, right? So it can able to cause the blue spots, right? So this is how we can easily identify that, right? Okay, so thank you. If you have any doubts, just ask me, otherwise we will conclude. No doubts? Sir, I think we can conclude, sir. Manohar, sir? Sir, I think we can conclude. Sir, I think we can conclude, sir. Class is over.
So thank you, Vimal Kumar. On behalf of our institute, once again, I am very much thankful and grateful to you for accepting our request and kindly and becoming available as a resource person to guide our students on the topic of fungal diseases of oilseeds. And you have covered all important aspects of the topic in a very comprehensive manner. And definitely, this will help our students in their preparations of ICAR, JRF, SRF, as well as NET ARS examination. So once again, I am very much grateful to you and thankful to you for guiding our students. Thank you. And dear participants, we will meet tomorrow in the morning session. Okay. And their morning session will be on microbiology, and afternoon session will be, I think, on entomology. Okay, we will let you know through the our WhatsApp group. So we will stop here then. Shall we stop, Vimal?
Okay, sir. Thank you. Welcome, sir.
Yes, thank you. And you will have to end the meeting, please, because you are a host.